We will first talk about an action potential. An action potential is a conduction signal that occurs at the trigger zone of an axon. The strength is always the same, and it's an all-or-nothing response, or in other words, it cannot be summed. The default charge or resting member potential is negative 70 millivolts. When it reaches negative 55 millivolts, it triggers the all-or-nothing action potential. It depolarizes because along the axon you have voltage-gated sodium channels and voltage-gated potassium channels. Voltage-gated channels open when the voltage changes to a certain number. So when the trigger zone hits negative 55 millivolts, the voltage-gated sodium channels will open and leak into the cell. Once the voltage reaches positive 30 millivolts, the sodium channels will close and potassium channels will open. Potassium will start to leak out and hyperpolarize the cell, and it'll reach past negative 70 millivolts. Sodium and potassium channels will reset themselves to reach the resting membrane potential by sodium-potassium pump. Next, we'll talk about the conduction of an action potential. Action potentials will jump from one node around VA to the next node around VA. Voltage-gated sodium channels will rush sodium into the axon. The surge of sodium with channels open creates an electrical signal that remains constant. The signal again will jump from one node to the gap to another. But once the action potential moves down, the voltage-gated sodium channels will block or inactivate, inhibiting backwards flow. Next, we'll talk about the release of a neurotransmitter. A neurotransmitter is released at an axon terminal. The voltage-gated calcium channels will open, causing the calcium to rush in. The calcium signals the vesicle to exocytose. The neurotransmitter then will be released from the vesicle into the synaptic cleft and will then bind to the appropriate receptor into the next cell.